And I'd like you to start with me by just imagining what it would feel like to be at a time where here you're taking care of patients with serious neurological disease. And imagine what it would feel like that we finally have a treatment for some devastating neurological illnesses like Alzheimer's disease, Parkinson's disease, ALS. We finally have something that will stop it in its tracks. And then think, what if we had the tools to actually diagnose at the very earliest stages so you can intervene and prevent that progression, or maybe even use things that could repair the nervous system. And that you have drugs that are so highly specific that go right to the target in patients who have a marker where you know the response rate is going to be extremely high and the risk and the safety is really reduced. And then because the high efficacy and the value of it, the barriers for access to that will also go down. And then finally, imagine that we're at a place where those who are underrepresented in research are now fully involved, and those that have been undertreated by some of these medications are beginning to experience the benefits of that. Well, to me, that's the future I'm looking for, and I think there's a lot going on in science that will gradually get us there. But I think we have to start with that vision, and I want to start with just talking about the pace of innovation and new technologies, and that's driving what's going on in drug development. If you think about some of the things, just to name a few, the high precision instruments, the use of things like 3D printing. If you look at genetic therapies, and here I have here big data, big emphasis on that in terms of real-world data, and things that we can learn from the amazing amount of data that's coming in and how we analyze that and how that can translate into things that improve care. And then this concept of personalized or precision medicine, and we'll talk about that a little bit more later, and the impact that that can have. I think it's, as I say, an exciting time. But I'd like to draw us from all the things that are going on in medicine at a very quick pace to things closer at home within the area of neuroscience. And for this, at Biotechnology. For this, I'm going to just talk a little bit about the birth of biotechnology and how this all started. This very young field, and you think about it, back in the early 1970s, Kurt Boyer, a geneticist, and Stan Cohen pioneered a new scientific field called recombinant DNA technology, and they met with a venture capitalist, Bob Swanson. And he was excited by the technology, and that was the birth of Genentech. And when I drive to work in South San Francisco, you'll see this sign up here. It says, the birth of biotech. Since that time, it's really exploded. All the companies around the world are using all kinds of biotechnology to advance us in science. And so it's an exciting place to be. And in fact, we celebrated our 40th anniversary this year. And the Smithsonian Institute actually did a whole exhibit on the birth of biotech and recognizing all of the different things from that. So it was a really interesting place. But one of the things that biotechnology does is it uses living material, cells, proteins, and one of the expertises and advances that are continuing are in the area of advances in antibody engineering. And I'm just giving you a couple examples here, because this is the next frontier, so to speak. Things like antibody fusion proteins and antibody drug conjugates, where things that we hadn't thought about before. We're talking about monoclonal antibodies that are very specific to a target, but now they're being engineered such that they have one arm that's specific for that target and another one that brings like a chemotherapy agent right to that target to not hit any other cells in the body, but just destroy the cancer cell. That kind of technology. There's some more. A variety of ways to engineer proteins and antibodies to help with evolving therapeutics. I have to tell you that all of these technologies are going to change the way and improve on the kinds of drugs that we're going to be able to develop for patients. So let's look at neuroscience. An example. This is with the brain shuttle technology. This is using an antibody engineering platform that was developed at Roche Genentech and looking to increase the penetration of large molecules across the blood-brain barrier. One of the problems we have in neurology is, in terms of drug development, the ability to use these very highly specific large molecules is getting it across the brain. And so let me just show you a little video, if I can get this to work, kind of explaining this particular example. Okay.
The ultimate unknown, besides space travel, is understanding the human brain. It is the most complex organ in the human body and probably the most complex system on Earth. There are 100 billion neurons in each of our brains, which is equivalent to the number of stars in the Milky Way. And there are 500 trillion connections in a single human brain. It's really the great unknown. I, as a neuroscientist, want to treat neurodegenerative diseases, and Mark's world is therapeutic antibody development. We focus on a single molecule and how it may be playing a role in disease in the human brain. But because of this barrier between the brain and the rest of the body, we were limited in our ability to get antibodies into the brain.
The blood-brain barrier is a significant challenge because antibodies and most molecules don't cross.
Right.
And so we were looking for some approach where you could piggyback on nature's systems to get our drug into the brain.
The problem is that no one's been able to do it in a setting that is really reasonable for developing a drug. The analogy that I like to use, being a skier, is it's like a ski lift.
The ski lift is the receptor or the pathway that we're trying to utilize to get molecules from the blood into the brain. It's always churning, going up and down the hill, right? And then you have molecules in the blood, which are essentially the skiers at the bottom of the hill. And the way that people previously had looked at this was they had very few skiers. And so in order for those skiers to get on the lift, they had to bind to that lift pretty tightly. But they were just getting stuck going around and around, and they never get into the brain. So the problem was, how do you get them to get off at the top? We realized that in a therapeutic setting, you're going to have a lot of skiers. And because you're going to have a lot of skiers, they don't have to be as fond of the lift. They just have to have a weak affinity, a little bit of encouragement to get on. And then when it got up to the top, they would fall off and be up at the top of the hill.
And that was a big idea.
I don't think this kind of work could have been done in other places in the world. It was the resources that Genentech provides in terms of antibody production, protein production, purification, our ability to make bispecific antibodies. We had to have one arm of the antibody that could bind to the receptor or the pathway, and the other arm that could do something functional to the target in the brain once it got into the brain.
In this case, it's a molecule that is required to produce the toxic peptide that causes Alzheimer's disease. Mark's lab then engineered the antibodies to bind weakly, and my lab went and proved in vivo that that engineering resulted in better brain uptake and better activity of the antibodies.
One of the nice things about this project, certainly we're trying to have an effect on this enzyme in the brain, but if we get this technology to work, you could take this approach and apply it to a lot of different medicines.
And in fact, that's our dream. And I think having collaborators that will be able to take risks together. It's a lot easier for me to take a risk when I know that Mark is like, let's do this. When the two of us agree that something needs to be done, people around us may think we're crazy, but we still do it.
That's just an example of, in neuroscience, something that's advancing. And that project is still advancing, and I think it'll be interesting to see what happens as we go into various areas, particularly in Alzheimer's disease, with that kind of technology. I'd like to spend the next few minutes telling you a little bit about how do you get from the big idea to the actual drug that you're able to give a patient in the clinic. And you may have seen a variety of slides similar like this before, kind of walking you through phase 1, phase 2, phase 3. But what I'd like to do is just briefly highlight a few things here and give a little bit of color and perspective on some of these stages. So I borrowed this slide from PPD, which is one of the CROs there, but it could have been from anyone because it's pretty generic in terms of its in the phases here. But it starts with the pre-discovery. Right? Big idea, the idea, the target, what are we going after that we're learning about? A lot of you were involved with this and with the drug discovery part, finding out something that may actually be the target or a good target for drug. I'll start with this first line, which you're probably familiar with, the fact that it usually takes about 10,000 to 15,000 compounds in discovery, going through the phases. Preclinical, you lose quite a few going from the discovery phase to the preclinical phase. Down to 250. By the time you get to phase 1, which is the first in human, we're down to about 5 compounds in it, and then going through phase 2, 3 to get to one that makes it out the door, right? And I can tell you it's been very exciting for me. I have been involved with, as a clinician scientist, with everything from phase 1, first in human kinds of studies, a very different, small number of patients. You're just making sure that it's going to be safe. And phase 2 studies where you're really looking at, is it impact, both proof of concept and proof of biological activity. So is the drug hitting the target you think, and is it having an effect clinically? Spend a lot of time here finding the right dose, all of that. I'm sure you've been involved in those kinds of studies. And then have been involved with that and with phase 3, which now you're looking at kind of seeing whether that efficacy is then indeed confirmed in a larger population, still within a clinical trial. And for this, have had the opportunity to lead multinational global trials with, thousands of patients involved with various different diseases to get to this stage. And then right now, I didn't mention the regulatory path down below, the IND, registering that at the phase 1 stage, and then at the end of phase 3, the NDA or the BLA if it's a biologic. And right now I'm involved with a program that is at that BLA stage, and I've been able to follow it from phase 2 to now where we're getting ready to launch in a couple of months, and it's been quite exciting to get to that point. It's something that will really be great for patients living with MS. The cost you see there to get through that, it's about $1 billion. Duration anywhere from 10 to 15 years. I think in the case of the one I'm working on it was 20, a little longer. But it's a long haul but an exciting place to be. You got to start there. And it doesn't end when you've launched the drug. There's always, in the post-marketing phase, kind of where I'm involved with medical affairs, you're looking to see now, outside of a clinical trial, how does this drug work in the real world, in patients, in the real world, and is it safe? And so we're continuing to monitor for safety, a key part of this, and evaluating for that, as well as educating. So a lot to do even in the post-approval phase of that. Another important aspect of biotechnology is not just the clinical trials, which is used with small molecules. When you get to things like very highly specific antibodies and proteins, the manufacturing is key. It's just as important as the trial design and the outcomes that you have in the clinic. And just a couple of words on that. So, unlike, I have a comparison here. You have aspirin with 21 atoms compared to an antibody, an IgG, with 25,000 atoms. The complexity comparison here is between a bicycle and a jet plane in terms of what it takes to make these kinds of molecules. And the inherent complexity involved, and the fact that it can take sometimes up to a year and a half just to produce the compound. So you can imagine what the timelines are in terms of that. In addition to that, these are grown and produced in living cells, and so there's interesting challenges. The benefit is you're talking about molecules that are very specific. They're not going to be going to hit off targets often. But there's also natural variation, so it leads to needing to control for that. And then you got to scale up. So it's one thing to make it in a small lab, for example. But now if you're really going to produce something that's going to help patients, you have the complexity of scaling up. And then this is just showing you in a single liter bioreactor where you can have more than 100 trillion cells that are working side by side, and this is showing you just how massive that is. In fact, today, back home, members of my team are visiting our manufacturing plant in Vacaville right now, just to get an idea of, these bioreactors are like 2 stories high. It's pretty massive, and an interesting thing to see the complexity in just making these kinds of biologics. It's kind of an overview of the process. So the end result in this, though, is the availability of some very powerful specific and treatments that might emerge from this technology. But what about, is everybody really benefiting from that? And we're getting these medical advances, but in fact, when you look at the participants in some of these trials, the African Americans, for example, representing maybe 12% of the US population, but comprise less than 5% of patients participating in trials. Hispanics, 16% of the total population, maybe 1% in trials. And when you look at diseases that are even more specific, the proportions are even less than that. And it's important to understand how these compounds and what we can learn from differences, as well as understanding how these drugs interact in certain patient populations. I'd like to just take a few minutes to highlight two diseases of interest. The first one being Alzheimer's disease on the older scale, and then multiple sclerosis that affects people in the prime of their life, young adults. And in both of these cases, African Americans are disproportionately affected by these diseases. This is a slide bar from the Alzheimer's Association. Not a surprise that this is, I don't have to tell this group, you can probably inform me on the impact of Alzheimer's disease on our society and the fact that African Americans are disproportionately affected by this disease. In fact, this is just a more recent study looking at an incidence. Sometimes you get cross-sectional data looking at the prevalence of Alzheimer's disease and dementia in general. This is one from the Kaiser Foundation in Northern California. They have over 3 million patient lives. And what was interesting about this is they looked at a time period between 2000 and 2013 at not only African Americans, but Latinos, whites, American Indian, Pacific Islanders, Asian Americans. And in all of these groups across the age span that you see here, the highest age prevalence was for dementia in this case was found in the African American population. Not a surprise, but substantiating yet again, not only the prevalence, but the rate of change is higher in this patient population according to this particular study. Another study, just to reference why, the fact that there is differences in some of the risk factors that may be contributing to this. Again, at an institution like this and a group, an ADRC that's studying this stuff, I don't need to tell you the fact that there are differences in what we say the risk factors, comorbidities, vascular disease, expression of ApoE, for example, and other markers. There's also diagnostic issues. One of the things that's key in trying to find successful therapies, especially in Alzheimer's disease, is to be able to intervene early. And in order to do that, you need to detect the disease as early as possible. And so challenges in doing so with some of the instruments that we use, how we go about detecting that, presents some challenges. And in fact, trying to figure out ways to increase participation. I think the talking to people here at the ADRC at WashU, they've done an amazing job of being able to include patients of ethnic minority population in research, actively in research. I think there's a lot of benefits. It's interesting, if you look at clinical trial work, just being in a clinical trial makes most people better. It's an interesting phenomenon, the placebo groups when you compare them. But I think that it's important to understand what that contribution can be. And when we look at multiple sclerosis, this is a disease, it's also looking at the parallels interesting, because with Alzheimer's disease, we're thinking it's primarily a neurodegenerative disease. However, we also recognize the fact that neuroinflammation is playing a role in Alzheimer's disease. And in multiple sclerosis, we also think of this as a primarily inflammatory, neuroinflammatory disease, but also appreciating the neurodegenerative component of MS. And so it's interesting that in both of these conditions, African Americans are disproportionately affected. When you look at MS, leading cause of disability in young people are relapsing remitting form affecting people ages 25 to 35. Much major cause of disability. And when you look at the racial and ethnic differences in the incidence of MS, again, I'm looking at another study here by a different group, but also looking at a Kaiser database, finding that the incidence of newly diagnosed MS was much more prominent in African American, 10.2 per thousand patients, versus Caucasian Americans at 6.9, and seemed to be less in the Hispanic Americans in this particular study. When you look at other populations, for example, the Wayland study, this was a study in veterans, you see the same trend with higher incidence rates of multiple sclerosis in African Americans. And the age of onset of the first symptoms also seems to be slightly older. Curious thing about multiple sclerosis is this geographic distribution. And what is that relationship? So we know this, the low prevalence near the equator. And the further you go away from the equator, the higher the rates of MS tend to be. And this is regardless of what race ethnicity is. And we know that migrants who move from a high incidence area to a low incidence area tend to have decreased risk of their MS, and vice versa. Migrants who move from a low incidence area to a high incidence area that's done at a younger age tend to retain the low risk in the origin country depending on their age when they move. We also see that in addition, that speaks to some environmental factors and possibly related to the vitamin D story, so it's an interesting one. But in addition to environment, the differences that we're seeing in African Americans is much higher than you see in Africans. Some of that's probably geography, but also genetics. And I'm just highlighting here a couple of genes where you see some differences that maybe underline some of the differences that you see. A couple other things to highlight. When you look at not only getting the disease, being more higher risk for MS in African Americans, you also see that some of the symptomatology and the clinical presentation is also more aggressive. And this has been shown in a couple of different studies: more rapidly progressive, larger lesion burden, more inflammatory CSF, for example, and mentioning things here from some of the other authors showing greater likelihood of spinal cord injury, of worse recovery from relapses. Very curious. So you wonder what's going on in the immune system there that may be leading to this more aggressive course. And this is just another paper kind of substantiating the same thing. And the reason I show these two examples is because in both cases, here with Alzheimer's disease and elderly populations being affected by Alzheimer's and low participation in trials, the same is true for MS. And the societies, both the Alzheimer's Association and here in the case of the MS, National MS Society, a lot of efforts are being poured into trying to rectify this low participation rate. And, however, have we been successful? Yes. So despite efforts, we still are not where we need to be. This is an interesting paper that looked at the period of time where there have been federal kind of mandates in clinical trials to recruit minority populations, and a report looking at, even still, we're seeing a disproportionate representation across the board in biomedical research. And ignoring this is a missed opportunity. The reasons for it are multiple, and this is another area where there's a lot of research that's going on here that looks at why is that. I don't put up here all of the reasons that relate to why there's low participation, but I did want to share this one interesting survey that was done that highlighted some of the attitudinal things, and I'm hoping some of this is things that we might be able to change. A couple of things to highlight from this were the likelihood of being thought of as guinea pigs, of not wanting to be experimented upon, are still attitudes that we see sometimes. I'm hoping, and I think that there's a lot of work being done to try to counteract this, and I think having more minority physicians involved in research is also an important part of that component. So bringing us back to what is it we can do to bring new technological advances, and how do we bridge the gap with not only minority populations. One of the areas that I think holds a lot of promise is this area of, we said personalized medicine, but it's really more precision medicine, where you're looking for clear markers of disease. Some of this may go along with race, ethnicity, some of it may not, but it'll be a smarter way to figure out how you can optimize therapies to particular targets, be more precise. And this is a release that, last year Obama announced a precision medicine initiative in his State of the Union address, and there's a lot of effort now to try to collaborate to make this a reality, both in enrolling patients across 1 million American volunteers in this effort, and the aim of which is to improve the ability to prevent and treat disease based on individual differences, lifestyles, environment, and genetics. Just more on the subject is, I think that translational piece is an important aspect of what we need to try to accomplish with this particular effort. And so not only is there an effort at the NIH level, but also at the FDA, there's an Office of Minority Health that's focusing on trying to improve the participation of minority populations within research. And a lot of it is being done on that score as well. I think this is a great quote that was shared by one of the physicians that was doing a lot of this research, is that the only way that we're going to make progress in decreasing the burden of disease is by understanding why it occurs. And we can't understand that without including diverse communities in our research. I think if I was to leave with a message of what I'd like, why this is important, this is why it's important. And I think that I don't have, this is just touching on some of the aspects from a medical standpoint, and why do we need to recruit more patients in the research that we're doing from a medical standpoint. There's what I'm not touching on, and what would be interesting to do at some point is have a wider focus where other things, societal changes that are needed, access to healthcare, other types of causes for this are involved, but I didn't really focus on that at this point. What needs to happen is for all of the areas of healthcare industry to come together to work on this problem. And I can tell you, as somebody who's working in a company, in industry, this is a very important concern for us, and something that we'd like to see progress be made in, not only at the trial level, but kind of beyond that. I'd like to finish with another videotape. And this one is one that is it's not really on biotechnology, it's more on bioengineering. But it's one that talks about the importance of innovation. And not all innovation is necessarily expensive. Innovation is really going to take us forward in a lot of ways. And it shows a group called Not Impossible Labs. I don't know if anyone of you have ever heard of that group. But it's a nice example of what we can do if we put our mind to it. So I'm going to show that and see what you think.
Daniel was 14 when the bomb took his arms. I read the article about him and had to help. I came to Sudan with 3D printers, laptops, spools of plastic, and the goal to build Daniel an arm. You ready? And then Daniel fed himself for the first time in 2 years. But it's never about just one person. You just double click. So this is the hand, 90 degrees. If we could teach the locals to do it themselves, the project could live long after we left. And it did.
Right? I showed that not because it's really, we have a lot of reason for hope. Technology is going to move us all forward, but we all need to be part of the process. And I wanted to show that because there's many examples I could hold. And I think that the time is now. The time is, let's get involved. Let's all of our community get involved with this. This idea of we're experimenting on people is really going to hold us back. I think if we're part of the process, we can all move forward together. And this was a great example of something that didn't cost a lot of money. I can't say that about the bispecific antibodies, but I do think that there's ways to figure out how to make progress. And I love the title, Not Impossible. It's that kind of thinking that's going to move us ahead.